Welcome back to my channel Knowledge Chronicle.
Today, we're diving into one of the most powerful tools in modern biology, recombinant DNA technology, the science of making hybrid life forms. By the end of this video, you'll clearly understand what recombinant DNA is, the exact steps we use to make it, the real enzymes that do the work, how humulin insulin is produced, real-world applications, and the safety issues everyone argues about. Let's get straight to it. What is recombinant DNA? Recombinant DNA, or rDNA, literally means DNA assembled from pieces that didn't naturally come together. Imagine taking a recipe page from a cookbook, inserting a paragraph from another cookbook, and then having a chef follow the mixed recipe. In biology, the recipe pages are DNA sequences. When we join DNA from different sources and put it into a living cell so that cell produces something new, that's recombinant DNA technology. Basic steps: cutting, joining, cloning. There are 3 core steps you should remember: cutting, joining, and cloning. First, cutting. We precisely cut DNA with enzymes called restriction endonucleases. Think of them as molecular scissors that recognize a short DNA word, for example, GAATTC, and cut at that spot. Different restriction enzymes cut different words and produce either sticky ends, those are short single-stranded overhangs that can base pair, or blunt ends, which are just flat cuts. Sticky ends are useful because they guide correct joining. Next, joining. Once you have a gene you want and a vector, and we'll talk more about vectors in a second, you join them using DNA ligase. DNA ligase is like biological glue. It seals the sugar phosphate backbone, so the 2 pieces become one continuous DNA molecule. Then comes cloning. After joining, we introduce the recombinant DNA into a host cell, usually bacteria, yeast, or cultured mammalian cells. The host copies the DNA and expresses the gene. Cloning here means making many copies, either many copies of the DNA for analysis and storage, or many copies of the protein the gene encodes. Important supporting steps include selecting the right vector, which is usually a plasmid, a small circular DNA molecule in bacteria, adding a selectable marker, like antibiotic resistance, to pick only cells that took up the plasmid, and screening to confirm the inserted gene is correct, by PCR, restriction mapping, or sequencing. Enzymes used: restriction endonucleases and ligase. 2 enzyme families do most of the heavy lifting. Restriction endonucleases cut DNA at specific sequences. They actually evolved in bacteria to cut invading viral DNA. DNA ligase joins DNA fragments together by creating phosphodiester bonds. There are other enzymes you'll hear about. Reverse transcriptase, which converts RNA to DNA. Polymerases, like Taq or high-fidelity DNA polymerases, which amplify genes by PCR. And topoisomerases used in some cloning methods, but honestly, the basics remain: cut with restriction enzymes and glue with ligase. So here's a real-world example: Humulin, the first recombinant human insulin approved for medical use. Before recombinant DNA technology, insulin was actually sourced from pig or cow pancreas. But then, scientists managed to isolate the human insulin gene, insert it into bacterial plasmids, and put those plasmids into E. coli. The bacteria, in turn, started expressing the human insulin protein, which could then be purified and processed into active insulin. That change, honestly, reduced allergic reactions and scaled up supply dramatically. This is textbook recombinant DNA at work: gene to vector, to host to expression to purification. So, where is recombinant DNA used today? Well, let's break it down. In agriculture, genes for pest resistance, like those in Bt crops, herbicide tolerance, or even improved nutrition such as golden rice, are introduced into plants. This can actually reduce pesticide use and increase yields. But it also raises some socioeconomic and regulatory questions. Now, in medicine, it's way beyond just insulin. Recombinant vaccines, monoclonal antibodies, gene therapy vectors, and diagnostic kits all rely on recombinant DNA. For example, the hepatitis B vaccine is produced in yeast, and there are many cancer biologics as well. And in industry, enzymes for laundry detergents, biofuel enzymes, and biomanufacturing are produced using recombinant microbes, making them cheaper and more sustainable than traditional chemical synthesis. Recombinant DNA technology is really powerful, but it's definitely not risk-free. The main concerns are, first, unintended spread. Engineered genes could transfer to wild organisms through something called horizontal gene transfer, though the likelihood does vary, and containment measures help a lot. Then there's allergies and toxicity. Introducing a new protein into food can cause allergic reactions, so rigorous testing is absolutely required. Ecological impact is another issue. Modified crops could affect non-target species or biodiversity if not managed properly. And don't forget biosecurity and dual use. The same techniques can be misused, so strict regulation, lab safety, and oversight are just essential. Regulations and risk assessments exist for a reason. Oversight, containment, and stepwise testing help minimize these risks. Saying it's completely safe or it's totally dangerous is just too simplistic. Each case really needs to be evaluated independently. Alright, here's a quick recap of key terms. Plasmid. That's a small circular bacterial DNA used as a vector. Vector, basically, it's a DNA carrier for delivering a gene into a cell. Restriction enzyme. Think of it as molecular scissors. Ligase. That's the molecular glue. Transformation. That's introducing recombinant DNA into host cells. And selectable marker, a gene used to identify successful clones. So that's recombinant DNA technology in a nutshell: precise cutting, reliable joining, and powerful cloning, all used to make medicines like Humulin, improve crops, and create industrial enzymes, while demanding really careful safety checks. If you want, in the next video, we can break down step-by-step lab protocols, like how to design primers, pick restriction sites, and choose expression hosts. If that's useful for your exam or project, just tell me in the comments. Your one like, share, and subscribe will help us create more valuable content.